According to the American Cancer Society in 2008, it is expected that 1 in 8 women will be diagnosed with breast cancer in the course of their lifetime. Breast cancer is the second most common cancer in women in the United States, and the occurrence of the disease has once again begun increasing. The most severe form of breast cancer is metastatic breast cancer. This type of cancer spreads from the breast tissue to a secondary location. According to Rubens and Mundy in the book Cancer in the Skeleton, the most common secondary site of cancer metastasis is bone, and once breast cancer cells colonize bone, the cure rate is almost zero. My interest in breast cancer led me to join a lab group last spring whose research focuses on breast cancer that spreads to bone. When studying breast cancer that spreads to bone, it's important to study not only breast cancer cells, but also bone cells outside of the body, or in vitro, by placing these cells in cell culture systems. It is difficult, however, to study cancer in bone using conventional cell culture methods because they lack complexity. So researchers in our lab developed a more advanced cell culture system that we refer to as the bioreactor. The bioreactor creates a very stable environment to study this disease. Today, I would like to explain to you the features of the bioreactor by showing you how these features exceed the limitations of conventional cell culture methods. I'll begin by discussing the basics of those methods. In conventional cell culture, cells are grown by placing them on the bottom of a polystyrene culture dish. They're provided with medium, which is shown in pink, that is conditioned with various nutrients that allow the cells to grow. The cells, as they grow, secrete waste molecules in addition to large proteins called macromolecules that are important for cell maturity. As waste accumulates, it's important to remove it from the cell environment before it becomes toxic. To do this, we perform a media change. When media is changed, the entire contents of the culture dish are removed. As a result, the cells experience a very drastic change in their environment that adds to a phenomenon called culture shock, which refers to the instability encountered by the cells. To add to this culture shock, those macromolecules are permanently removed and cells are inhibited from maturing. So as you can see, conventional cell cultures create a great amount of instability and to this date have only been able to grow a single layer of cells in culture. Now the bioreactor has features and principles that overcome these instabilities. The bioreactor was developed by a former PhD student in our lab, Ravi Durjati, and based on a patent by Dr. Erwin Vogler. The bioreactor is a compartmentalized cell culture device. It's composed of 4 stainless steel rings secured together by 6 stainless steel screws. This assembly forms 2 compartments or chambers, the cell growth space and the medium reservoir. These chambers are separated from one another by a dialysis membrane, which is a semi-permeable membrane, and access to each of the chambers is through taper ports. The bioreactor operates on a principle developed by Gigi Rose of simultaneous growth and dialysis. Cells are grown in the bioreactor only in the cell growth space. And the cell growth space has a much smaller volume than the medium reservoir. So as waste accumulates, these waste molecules are allowed to diffuse into the medium reservoir. The semi-permeable membrane is impermeable, however, to the large macromolecules secreted by the cells. So by allowing those macromolecules to remain with the cells, the bioreactor creates a stable environment that allows those cells to mature. In addition, when media is changed to remove the waste, only the contents of the medium reservoir are removed. So this means that cells in the cell growth space are left undisturbed and don't undergo that drastic environmental change. Therefore, culture shock is minimized, and we have an even more stable environment. In fact, the bioreactor is so stable that we've been able to grow between 6 and 8 cell layers using this device. So now that you've seen sort of the intricacies of the bioreactor design, I'd like to briefly discuss some of the research breakthroughs that have been made in our lab using this device. When studying breast cancer that metastasizes to bone, it's crucial to model bone as accurately as possible. This is difficult because bone is such a complex tissue and only forms correctly when multiple bone cells interact over time. Because the bioreactor can grow multiple layers of bone cells over extended periods of time, we've actually been able to grow calcified bone tissue using this device. You can see in this image here a bone chip that was recovered from the device, and we've been able to calculate the density of that bone, and it's calculated to be almost as dense as actual bone. Now we can take this three-dimensional bone tissue even further and challenge it with breast cancer cells. These images come from a most recent publication in our lab by Venkatakrishnan, and they begin to show the process by which cancer cells metastasize to bone. The bone cells are featured in red, and the cancer cells are featured in green, and you can see sort of three main steps to this process. First, cancer cells adhere to the bone tissue. Then they begin to penetrate that tissue, and finally they replicate within the tissue and organize themselves into a very characteristic fashion. And this is something that's never been done before, so this device has really created many advancements in our research. My research takes this model a little further, adding chemotherapeutic and osteoporotic drugs to this cancer and bone model to develop a treatment for this disease. So as you can see, the bioreactor has really aided in how this process is studied. So today we've taken a look at the intricacies of the bioreactor design and how this device is being used in metastasis research. Ultimately, the stability of the bioreactor creates an excellent model to study breast cancer metastasis to bone. I hope you're able to appreciate the implications that this device has on the future of cancer and bone research. It is my hope that with this technology, my research will lead to a breakthrough as well. Thank you.